First of all, I introduce myself. I'm Nima. I will tell a bit more about my background. And as Girly mentioned, please feel free to ask questions. It will help to have a more dynamic and interactive discussions. So I chose this topic, the unseen in omics, lessons from non-significant results and biases. And before we start, I say a few words about my background. So there will be a few questions which I will ask you, and you can answer through Mentimeter. So just use your mobiles, just scan the QR code, and then you will answer some questions. Okay? So believe it or not, I have received this question whether I am Nemo, the fish. But I'm not the fish. I share some genes with fish, but I'm not Nemo. I'm Nima. And I'm originally from Iran. I did my undergrad and master's degree in Iran, the University of Tehran. And then I received a scholarship from Erasmus and moved to Europe. The first year I was in Netherlands, Wageningen. Is anyone coming from Netherlands or have been in Wageningen? Okay. Alles goed? Yes. Oh, okay, good. Yeah. Oh, perfect. So then for the second year, I came to Sweden and I did my degree project with Leif Andersson's group, which I will tell you a bit more about that. And then I continued on the same project as my PhD project, and then did a short postdoc in Leif's group. Then I continued with the second postdoc in Åsa Johansson group, where I did my where we did a whole genome sequencing data analysis in 1,000 Swedish individuals, and we tried to find the association with proteomics data. Then I started 2019. I started at NBIS, and I received my first and last research grant as soon as I signed the contract with NBIS. So I couldn't do the work actively, I mean, but we managed to do some long-read sequencing data, and it was published a few months ago. And I will tell you a bit more about like my role, which might be one of the career paths that you may become interested to take in future. So I give you a few examples then to have a picture that different things that one can do. For my master's degree project in Iran, which was a collaboration with Minnesota University, I developed a statistical model to predict the probability of abortion in dairy cattle. And I managed to develop a software and delivered it to one of the biggest dairy farms in the Middle East, at least at that time. Another activity was outcome of one of the projects during my PhD, where I worked on a skeletal disorder in Shetland ponies. It was also quite messy. I will tell you details in upcoming slides. But the outcome was a genetic test that I designed and has been patented, and it is in the market. Breeders can use it both in US and also Europe. And for that, I received some legal advices from Uppsala University Innovation. Another startup was together with two other friends and colleagues. We worked on QC of agricultural products using DNA sequencing, which was very successful in the beginning, but unfortunately because of Corona, we couldn't scale it. So it is now frozen. Something which I have recently started is another startup to help the applicants who are trying to find they want to land their job in bioinformatics and data science. It is called a Skills Unfold. It is an ongoing activity, so if you are interested, you can get connected through LinkedIn. There are newsletters with tips and tricks about CVs and the related topics. I had also a workshop on the same topic, which you can watch on YouTube now. You can just scan QR code and then watch it. Before we move on to the real lecture, I have received this question from freshly graduated students or even senior researchers who want to switch their careers in bioinformatics and data science. So I try to dig in a lot and find like a good resources to share what are the job titles like bioinformatic analyst 1, 2, 3, which is more common in US. But I thought maybe I can get more accurate results from the community. So active professions and also students are filling in this questionnaire, and it takes very few minutes. So it eventually can be something useful for your future as well. And the statistics, I'm planning to publish it through the Skills Unfold. So feel free to fill in the questionnaire, and then you will receive the results. Now, NBIS, National Bioinformatics Infrastructure. Sweden is one of the platforms at SciLife Lab where I work. We are more than 130 staff. We are distributed in different nodes, Umeå, Uppsala, Stockholm, Linköping, Göteborg, and Lund. And I think in the team that I'm working, cell and molecular biology, most of us are sitting in Uppsala. So our core activities is to help Swedish researchers in order to build the knowledge analyzing omics data, and so streamlining the bioinformatics. We transfer the knowledge to the team, so in upcoming projects with the same data type, they will be able to do the analysis themselves. And we do that in 3 different tracks. By having hands-on on the data analysis, which will be different modules, like we have peer review, we get 500 hours support for free. There are user fee track as well, where users pay 800 Swedish kronor per hour. And partner projects, which I am doing one of them at the moment, is interaction between NBIS and spatial biology. So they pay 50% of my salary to help them establishing new workflows or doing data analysis. The next track is computational resources, tools, and guidelines. We have different teams, data management, system development group, which they work on developing websites and portals. And we have 2 tracks activity in relation to the partner project that I have. And training, we have a lot of courses. Especially for PhD students and also postdocs. Occasionally we have had master students as well, depending on their status and whether the PI have been able to support them to come to the course. You can find all the courses we have on the SciLife events and NBIS courses. And with that, I think now I ask you a question. I know you have different background. You are going to do a program, advanced research training, right? Yeah? Okay, so I would like to hear, how do you see yourself in coming years? Degree project, PhD, industry. Few more seconds. And then we move on. But what I can see is that most of you are interested in doing a PhD, which is good, and some of you are interested in the industry, degree project. Okay, cool. Shall we move on? Now, after lectures today, you will change your mind. No, I'm joking, of course, but it's good to know. So this is the outline. I will tell you a bit about biases in science, and then I give you examples of the biases in different projects, like Atlantic herring, where the first project is where we didn't have the reference assembly, and then I developed a workflow to go from exons to non-coding regions where we can capture more variation. And then sex chromosome discovery using literally junk data. I asked Gerli, you have worked with NGS data, and you know how to do work with NGS data and their reference genome assembly. Yes? I see some nods. Okay, I take it as yes. Because it will help when it comes to explaining the bias there. It took 7 years to publish it. So I'm saying that because it may happen to you, so don't give up. And the skeletal atavism, it was also in the most complex region of the genome, chromosome unknown, which does not exist anymore. I will tell you about that as well, which led into a genetic test, so you can also expect some of the unexpected outcomes as well. How to reduce biases, and the last part, it was biases beyond science. So I won't be talking about the problems in the science, but beyond that, as individual level, when we are going to interact with other humans, other individuals. I had planned to put, since I'm obsessed by myself, I will repeat about me at the end. No, I'm joking. It's a mistake. It was just an in silico mutation. It's one of the concepts that you will deal with. I mean, we don't use this title, this expression when we do science, but maybe not only in science, in our daily life, we may also do that. And so 3, 2, 1. Okay. No idea. Good. To reduce complexity, yes. Reducing the amount of mistakes. That's also a good point. So, yes.
I was thinking it's when 7 years of work ends up in a supplementary on the publication.
Yeah. That can also be, yeah, exactly. So, yes, that was a good point. That's also a true part of that. I mean, doesn't matter how much time you spend on it, so it may not give you as you expected. But sometimes doing a standard workflow with a proper study design will give you very good outcome. It really depends. So thanks for your inputs, and now here we go. So it's bottom-up approach or reductionist approach. We try to break the problem and try to do it in very small mini tasks in order to have a better understanding and have a bigger picture at the end. So when I'm talking about that, maybe we can also represent it as the iceberg. So what we see is just part of the iceberg, which is on top of the water. But we want to characterize and understand what is beneath it. So if we take the tip as like a trait, a phenotype that we are trying to study, we try to read that by different data sets. Like we study the genome to identify underlying factors leading into the trait or phenotype that we can measure. Or we do transcriptome or epigenome or proteome or many other layers of information. So we go one by one. This is like a reductionism. So we also try to limit, to control the. It's supposed to be 240 minutes. Anyway, so I guess now you know what's the point of reductionist approach, right? Yes? Okay. So with this in mind, I would like to tell you about Human Reference Genome Timeline. 2001 was the first draft human genome, which was a very big and ambitious project. Many researchers and labs were involved across the world. And there were 5 individuals used in order to build that map. There were a lot of gaps in the genome assembly at that time. The next step was to characterize the genomic variation across population, different individuals, and HapMap project aimed to address that, to identify common genetic variants. Then from 2003 and 2012, we could see the birth of many other layers of information like epigenome, transcriptome, to give us better understanding on the biological mechanism we see associated with given traits or diseases. 2012 till now, we see the appearance of nation-specific genomes like Swedish genome or Chinese genome or Korean genome. It helps us to identify population-specific variation which are missing from the first draft genome assembly. By advances and also innovation in the sequencing technology, we can now fill in the gaps and even go from telomere to telomere. This was also one of the very interesting researchers recently published from 2022. This is recent. It's a gapless genome. In addition to that, we see the appearance of pan-genome projects. So it's not only one single genome. We can generate genome for every individual, which can have very unique and specific variation. So the question is that does reference genome assembly represent all the variations? If you look at the Swedish genome assembly project, which was the researcher used long-read sequencing data technology, PacBio, and it was done at SciLife Lab, just a few meters away from here. They sequenced and assembled genome of 2 individuals, and they identified 10 million base pair which was missing from the reference genome assembly. It's fairly large. It's larger than expected. And interestingly, 6 MB of this 10 MB showed similarity to what was discovered in Chinese genome assembly as well. Using 200 individuals, they mapped it back to the Swedish genome assembly, and they could identify 10,000 false positive variants. So imagine that we are working on a different set of variants, and then with this new picture, we can use more reliable variants to do downstream analysis and also identify completely new variants from the part of the genome which was missing from the first draft. And when we talk, you know, you have seen Manhattan plot, and I think you have generated it yourself as well. So this shows us non-significant results. And here we see some peaks showing significant results. Even when we have significant results, what's the chance that the variants just at the border of the threshold that we put are not useful, does not telling us important information? We use this cutoff because of just correcting for multiple testing. But if we increase the number of samples and try to adjust our analysis, this line may move. And we may, in another study, we may see, okay, these guys can also be significant. So what I'm trying to tell you is that what are we deciding? We may unintentionally have some biases in the data analysis. So, and we try to look for low-hanging fruits. That's how we do it. And it is, again, a bias or reductionism that we have in our data analysis. Atlantic Herring Project. If you're Swedish, you have heard the name strömming, surströmming, seal. Has anyone tried surströmming? Yes? Okay. Yeah. It is in my list to do. I haven't dared to do it yet. So it's culturally and economically very important species for a Scandinavian country, especially Sweden. One of the main characteristics of this species is the large population that you see here, and the killer whales are feeding on them. And if you go back in the history, this is a woodcut from 16th century. One of the key characteristics that you can see here is the dense population of the fish. And during spawning time, you could even leave an axe in the water and it would stand because it was too dense that the fish would keep it in the place. So, but due to overfishing within the last few decades, we could see reports about the collapse of the certain populations. And the idea of this project was to characterize different populations in order to have a better management, fishery management. One of the other characteristics of this population is, again, related to the massive population that they have. When it comes to spawning time, they come to the shores and leave a sperm and egg. The white color that you see is like a pool of sperm that has changed the color of the water. And so it's also one of the key questions that we had, what is the sex determination mechanism in this species? The silver of the sea is also another name that they use to call a herring, and in northern hemisphere it is labeled or categorized based on the geographical distribution. You have Pacific herring, you have Atlantic herring, you have Baltic herring. In addition to that, spawning time has been used to label the populations. Some of them are spring spawners, some of them are autumn spawners. And in the data collection, which goes back to 1980s, there were 20 or so population collected from Gulf of Bothnia, where we have low salinity, 3 ppm. In the south, where you see the salinity increases to 6, 12 ppm, and when it comes to oceanic water, where we have 35 ppm salinity. So these fish can sometimes migrate and also feed in different salinity. So it has very interesting another interesting characteristic of this species. So the task for my supervisor and teams before him, Leif Andersson, was to understand what are the differences between these populations. With the technology at that time, he tried to characterize 17 loci in different populations, and as you can see, these shows different forms of proteins, isozyme, and then trying to see whether there is a specific differentiation between the populations. But as you can see, there is overlap between all of them. You cannot identify any unique or private variants or variation for each of these populations. So later, by use of advanced technology sequencing, there were some reports that they could identify a handful number of variants or microsatellites showing differentiation between population, but it was not possible to relate it to any functional element of the genome, no genes. So to do that, we needed to have a good, high-quality genome assembly. We didn't have that. And we tried to generate a transcriptome assembly. This is one example of that. And then I mapped the RNA and DNA. This is the exome assembly pipeline that is different from exome sequencing. And I explain it in a few seconds. So after mapping DNA and RNA, so the RNA is shown with the black line. Since it is transcriptome, we expect to see continuous mapping. But when we map DNA, since some of the reads go into intronic or promoter region, we see a dip. So the dip that you see here for the DNA are potential borders of intron-exome boundaries. So what I did, I extracted DNA sequences that have been aligned to this region on this gene, on this transcript, and also reads that did not map to this transcript. And I ran mini assemblies. I think we had 30,000 transcripts, and I did 30,000 assemblies for each of these transcripts. And this is the outcome. So the green shows the potential exons, and the black lines shows the intronic or promoter region. So it shows that we could move from exonic region to non-coding regions, which gave us more space to align the reads and call variants and identify differentiation between populations. As a test, I checked whether these non-coding regions consist of promoter or not, and it was consisting of a promoter region. So this is something similar to Manhattan plot. These are the variants from the exome assembly, and as you can see, we have some peaks, but it is scattered, right? So we cannot see how large is this block. Now imagine if I map it when we have the genome assembly. When we map it, we can see, wow, it's not only handful number of variants that we could see show a differentiation between population. What we see is a large block. There are many variants. So we are not talking about few number of SNPs. We have a large block which can represent haplotype differentiation between the individuals. Any questions so far? So jump in if you have any comments or any questions. So now with having large population, large genome assembly, we could characterize many more populations. So what you see here is a Pacific herring, which shows quite large divergence compared to Atlantic and Baltic. And if you zoom in the Atlantic and Baltic, what you see here is like a representative, or like a populations that belong to the same. The genetic differentiation between them is very minor, is very small. But the picture that I showed you, the Manhattan plot that I showed you in the previous slide, yeah, I think it should be for salinity. But if we group the populations living in the saline water or brackish water, we could identify differences in this region. Any questions so far? Sorry, I don't know your name. What's your name?
Andreas.
Andreas asked an interesting question, whether the identified novel sequences from Swedish genome are scattered across genome or coming from given regions. It is scattered, spread different chromosomes, and when it comes to chromosome unknown, I can say more specifically from my own experience. But I don't know exactly like which fraction of which fraction of the novel sequences are coming from given chromosomes, but it is scattered because across the genome we have different complexities. Some regions you have more repeats, and it's more likely that you have difficulties to assemble. And one example in the herring that I had, I didn't have this slide, but one small fragment of the sequence was located towards end of a scaffold. Scaffold is like a step before mapping it to chromosome level assembly. And when we have usually at the end of the scaffold is the place that you have misassemblies. So it's usually ignored. But what I found was that that misassembled region had a massive copy number variation from 2 copies to 100 copies, and we validated it in the lab. We could see that it has a negative correlation with the salinity. So if a fish is living in low salinity, needed to have more copies of that gene, which is called HCE or hatching enzyme. So it helps the hatching the egg during spawning and also in given salinities. So with advances in sequencing technology, when we have longer reads, we can go over the repeats and be able to characterize or identify more regions. Yeah. Now that we talk about diversity in the populations, one of the key questions in evolutionary biology, which is called queen problem in evolutionary biology, is sex determination. And it has quite diverse forms. You have a genetic sex determination, where the genetic dictates what will be that species, that individual, will it be male or female. Sometimes you have environmental factors, which the temperature, for example, in some turtles and some fish can determine the sex or influence the sex of the individual. And sometimes a combination of both. The most diverse one is observed or reported in teleost fish, and Atlantic herring is a teleost fish. So let's look what's the story here. So it's a multi-omics approach, a project that we use different layers of information: genomics, transcriptomics, proteomics, and also functional validation. I'm not going to walk you through all of the steps, but these are the information that we use to characterize the genes involved in sex determination. The first screen that we did resulted in a flat Manhattan plot. We didn't have any significant association or regions to differentiate sexes. So then I got the task to look into this further, and then I analyzed allele frequency differences by chi-square. And this was the first picture I found. It was a large block in the middle of one of the scaffolds. It was still a scaffold version of the assembly. And I was very excited. because I could see a very significant difference at the allele frequency level between males and females. We had 8 individuals, 8 females and 8 males, and then I tried to look what is the haplotype representation in that region. So the AA allele is shown in gray, is like a reference allele genotype, and then the light blue is heterozygous, so positions that have a copy of A and B, and BB is where the individuals are homozygous for alternative allele. So the females show that they are fixed or homozygous for reference allele. And surprisingly, we thought the reference individual was a male. But when we look at genotypic data, we can see it resembles females. So maybe we are looking into female genome assembly. This is also another biases that we have in this case. Sometimes it's difficult to determine the sex of herring when they are outside of a spawning time. Now let's look at the males. So males mostly have heterozygous status. So one copy similar to female, one copy unique to males. There were some individuals that we didn't know their sex, but what we see is combination of both of these forms. So one of the conclusions from here is that we have identified a region that males are heterozygous, like XY form, while males are homozygous, like XX, that we have in human as well. So then I genotyped 200 individuals, and I could see 88.4% consistency between genotype and phenotype, which was very good validation of the findings. So take-home message here: males are heterogametic. Sex-associated region is located on the region that I showed you, that we could map it back to chromosome 8. Shall we call it chromosome X or not? We will continue and see about that. The key question: any sex determination gene? So I was very excited. I said, okay, maybe there should be some sex determination gene, and then we are ready to publish it. So this is a region, and this shows the divergence fraction. It's just level of showing the level of differences, genetic differences between males and females. You can see there is a difference within the region, sex-associated region, and the surrounding region, which we can call pseudo-autosomal region. It is interchanged. The recombination happens between males and females. But in here, the differentiation is quite distinct, suggesting that there is no genetic material exchange between these two regions, within this region between two sexes or chromosomes. So none of the genes within that region were associated with sex determination cascade. So I was a bit disappointed, and we were wondering what to do. So I went back to the mappings. So if you have paired and reads, this is like I call them happy mates, that they are sitting together. It's like a tango. They can have their locations, and within the sex-associated region, we also have happy mate. But sometimes we can identify reads that are mapping on the breakpoint of the sex-associated region. We see a clip, soft clip. It is called soft clip. So the mapper tolerates some bases not to map. So it is labeled as soft clip. Sometimes we have unhappy mates or orphan reads that one of the reads are mapped, the other one, the read is not mapped. So what I tried to find was at which region I see enrichment of these orphan reads. And I could identify these. It was like a large fraction of the reads. All of a sudden, you have this randomly scattered across the genome. But when it came to this region, I could see, oh, I have elevated level of these reads. I could see many reads showing this pattern. So it indicates, what do you think, what's the reason? Any guess? Why we have reads like this, that one pair is aligned, another pair is not aligned?
Because this region is not present in the reference.
Yes, exactly. Yeah. So the task was to identify missing sequences from the genome assembly. What I did, I collected these reads together with unmapped reads and reads that one of them is mapped, and then I assembled the genome. And I identified 10 contigs, and one of the genes I identified was a sperm cation channel. So bingo, it was associated with the sex, but it was the secondary trait. It has to do with the sperm and gametogenesis, not the sex determination. I told you the story. This is called junk data, but labeling it junk data does not necessarily mean it is useless. This is another example that using unmapped reads and junk data can give us some important and valuable information. So what I did later, after doing the assemblies, we had a better assembly, and we had sequences that were like a chromosome level assembly, and some reads and sequences, contigs that we could not map to given genome. So then I compared the depth, whether are there regions that I see only in males and absent from females or not. So here was one example that you see. It is chromosome 8, and there are a bunch of them here, and unplaced scaffolds 2 to 9 and 18. So don't think about the namings. It's just the sequences that do not belong to any chromosome. So then I try to find the similarities between these unmapped scaffolds and the main chromosome, and I could see some similarities. It's not very visible. Do you see the lines? So it shows that overlap. We see this sequence from chromosome 8 exists in unplaced scaffold here as well, and it shares the sex-associated region. But the beautiful picture that we see is that this segment which was missing in the chromosome 8 or chromosome X, and when we compare the males and females, we can see males have one copy and females have no copy of this region. So this region is male-specific region. These should potentially have the genes determining whether this individual should be male or not. So when I was doing that, so it was 7 years of work, and another group came and published it. And I was very stressed, and I said, oh man, I spent 7 years, and I should have published it earlier. But guess what? They could beautifully show this nice peak on chromosome 8. So it was the first signal that we found. And they didn't report any sex determination gene. So it made me happy and say, okay, so I can still have my own story. So the story is like this. We have a copy of gene which is called BMPR1BB, and it is a transcription factor, receptor, known to be involved in sex determination mechanism or pathway. So almost 2 million years ago, this one was duplicated on chromosome 8. Over time, there were some mutations, which changes the structure of the this gene, made it BMPR1BBY, Y version of this. And then, over time, 15 million years ago, we can have the evidence that this gene, CAT-SPR3, which is a cation sperm channel, has been duplicated on chromosome 8. And CAT-SPR-G appeared sometimes. We don't know the date when it happened, but it was beneficial for males to have this gene, because if it was not beneficial, it will be excluded during evolutionary time and natural selection. And then, since we had one copy of CAT-SPR3Y, this gene, the autosomal copy, went through pseudogenization, so it became inactive. So I just touched upon very important concepts in evolutionary biology just within this slide, which happened to form the sex determination region in herring. So imagine this is one bias which is mix of scientific experience and also non-scientific experience. Imagine someone comes into the lab and say, I found a gene which is 60% smaller than you expect, but it is functional. So there were a lot of ha ha hoo hoo. It's not. It's wrong. It doesn't work, etc. But by validation, we could show that this truncated short version of the autosomal copy, which was duplicated on the Y region, is functional. So if you look at this structure, this is autosomal copy, and this is a Y copy. It is very truncated. You do not expect this to be functional. But I'm not going to bore you with the details of this experiment that our colleague did in France. It shows the activity of the BMPR1-BBY, these two, what you see in the bottom, together with other members of the this pathway. So this one shows the base level of expression, and by including and excluding other players, we test whether we see any specific activity of our gene of interest or not. So if we include the Y copy, we see that in absence of the autosomal copy, we see higher expression with both forms. And if we exclude all other members of this pathway, this and this, you see we still have activities. So what it tells us, despite of short size of this copy, it still maintains the enzymatic activities. And considering its role, it suggests that this gene may have potential activity to induce testis development. Is it clear or it was confusing? It was not. I can explain it again.
It was fine.
It was fine.
Understandable, yeah.
Okay. Okay, so the lessons learned from this part, incomplete or biased reference genome assemblies limited us to to identify signals of adaptation. And when we got the reference genome assembly, we could expand it, we could identify many more variants showing differentiation, or we could associate it with adaptation. And also characterization of the sex determination region wouldn't be possible unless we zoom out, look at the junk data. And refining genome assembly to capture genomic signal is one of the take-home message from this project. Skeletal atavism. So for any questions? No, crystal clear. Yeah, I can explain this to anyone, to everyone after leaving this room. That's good. Okay. How many of you like horses? How many do you ride horses? No one? Oh, okay, me neither. So for people who have a strong interest, who are very interested and fond of horses, this story can be interesting. So what I'm showing you is the X-ray of the normal development of the bones, where you see the development of the skeleton in the healthy individuals. But if you look at unhealthy individuals, you see the growth is quite different. There's an abnormal growth, and it leads into deformities of the skeletons. And if you look at the horses with these deformities, they cannot walk properly, they do not have complete development of the limbs, and they are usually euthanized within the first week of their life. So from different perspectives, animal welfare and economical impact, it is very important to understand what is genetic factors underlying these deformities. The first time it was reported in 1950s in UK. The inheritance status was reported to be autosomal recessive mode. And what is atavism? When a phenotype appears again after being excluded during evolutionary time, it is called atavism. For example, supplementary nipples in human or hind limbs in whales. And this is also an example which was in the during the evolutionary time, it was not beneficial to have, but after many years, it appeared again. It is called atavism. Does it make sense? Yeah? Okay. So, another example. We did GWAS with affected carriers and controls using genotype SNP chip of 50K. SNP chip. And no significant association. But when we did NGS data, we sequenced 6 affected individuals and a pool of controls. Do you know about pool-seq? Okay, great. So we have a pool of controls, 22 males with no affected offsprings, and with 55x. And here we had low depth. At that time, it was not very low depth, because these days you can also sequence with 1x and still deduce the result, deduce the genotype. So we had Swedish individuals. All of them were Swedish horses. So with this we could find, but not with the SNP chip. Anyone can guess why? So, do you know what a SNP chip is? It is a collection of the sites across the genome. So it is much cheaper than doing whole genome sequencing, because with whole genome sequencing you can characterize almost every basis. But with the SNP chip, we have the collected version. In this case, it is 50K. So we have selection of 50,000 SNPs distributed across the genome, and we did not find any signals. And one of the reasons that we did not find any signal is that the associated region was not represented within this SNP chip. So that's why when we did the whole genome sequencing data, we could find the associated region. So I want you to have your full attention here, because it's a bit complex picture, but when you understand it, I hope you appreciate the beauty of that. Not because of the colors. Yes?
How do you determine the SNP chip? Is it random, or how do you determine?
That's a good question. So it depends. So if it is custom SNP chip, so for example, for herring we did that. We say, okay, when we do whole genome sequencing, we could identify given genes showing differentiation between populations. So we specifically selected for informative genes, the informative variants. But sometimes you can also capture common variation in order to make a comparison between the SNPs are under selection and the SNPs which are not under selection show a Hardy-Weinberg equilibrium. So it's usually mixed. But if you have a custom set, you can select which variants to order or have in the SNP chip. That was a good question. So the beauty of this is not in the colors, but is in the signal. So here we have 6 individuals, and the bars shows in 1K window the copy numbers. So here is the region that we have 2 copies, and within this region we have approximately 1 copy in these 3 affected individuals. But if you look at the other 3, we see no signal, as if it is absent. And for these guys also, we have only 1 copy, so suggesting that there is one normal copy, but another copy is deleted in this region. And if we go forward a bit, in the downstream, we see that this region is absent in all of the samples. So it shows that there is a second deletion. But if we compare it to the control pool, this is the z-score depth. So when it is close to 0, it shows average expected depth of coverage. And here we see more or less we have almost 0, but at least we have some depth. And in general, we may have a bit of bias coverage here as well, because of gaps and complexity in this region. So this is the track that I want you to draw your attention. These large blocks are gaps. We do not have any sequence. We don't know what are the bases within that region. And I take the analogy to the black holes in the genome assembly where it stores information, and we will see whether it has information or not. So take home message from this slide. We have 2 deletions, 1 large deletion which is completely absent in some of the individuals, and in the other individuals we have both copies, one deletion 1 and one deletion 2, which is shorter, and one deletion 1. Pay attention to these two genes. The del1 removes shocks, which is in the del1 region, and then CSF2RA, which is downstream. There is no deletion, but just keep in mind these two genes. Any questions so far? No? Okay. Using PacBio sequencing, long read sequencing, we could. The one thing which I wanted to tell you is this is chromosome unknown. So here I have a soup of contigs coming from different chromosomes. So here I have sequence from chromosome X and Y, so the autosomal region. Just few kb downstream, I have regions from chromosome 21, 29, chromosome 17. So it's very mixed, and it was a pain to put these together. So to do that, I had signals from 67 MB. This is I think 20 something, 26 MB. And how can I put these two together when they are away 40 MB from each other? So to do that, we did PacBio sequencing, and then I could assemble everything within 3 contigs. This is contig 1, contig 2, and contig 3. So in contig 1, again, I mapped all the sequencing data. So here we can see the representation of the del1 region in these 3 cases, which they have 1 copy, and this is the normal pool, which we see more or less the same number of copies as expected. And then for the other 3, we see deletion, and here we have shocks when we compare this contig to the human chromosome X. In contig 2, we see the same pattern for the del2, but there is no coding gene when we map it based on homology to the human chromosome X. And in downstream, this is where we have the another breakpoint of the deletion, and CRLF2. Do you remember CSF2RA gene, which was downstream of the deletion? This gene was in the gap region. It's here. We didn't have it. It was in the gap region. We didn't have any sequence to characterize it. So take-home message here is that the deletion not only deletes shocks, but also CRLF2. So here again is another bias. Absence of evidence is not evidence of absence. So we didn't have that in the SNP chip, but we continued. We did whole genome sequencing data. When we did that, we could also improve the picture that we have by including identifying a second gene deleted within this region. Even with PacBio sequencing at the breakpoints here and there, we had a lot of repeats, which were limits to identify the true breakpoints of the region. Take-home message: high complexity of the region, repeats high GC content, as I told you, limited our power to sequence and characterize the region. A SNP had not covered the identified region within a SNP chip as discussed, and this is also interesting. When I met the vets, when the breeders had a meeting with the team, we had a plate, and we calculated the allele frequency based on the plates that we had, based on the samples that we had, and we overestimated allele frequency. When we talked with the breeders, they said, no, you are giving us wrong information. And they were right, because we had a biased sample. We had included more carriers, more individuals that had affected offsprings. So we calculated the wrong allele frequency in the population. So this was also another bias which may happen to you as well. So how to reduce bias? Not much left, actually. One approach is to do pan-genome. And it is fairly recent that that's the task, the sort of result Human Pan-Genome Reference Consortium reported recently. 47 genomes, mostly representative of African individuals. And now we can capture more population or ethnicity specific variants or sequences, which would potentially be missing from the reference assembly. Another effort is a large genome project such as Zonomia, where they sequenced 240 mammals. It was also one of the projects from INBIM, the department that I'm working. Shashtin Limbert team also did that. The first version was 29 mammal, almost 10 years ago, and they increased it to 240 species, mammalian species. The idea here is that to identify regions which are completely conserved, and they called it evolutionary constrained regions. These are the regions that show slow change in the DNA. So these are evolutionary, very important to maintain the structure as it is. What happens if there will be mutations in this region? They try to show that by looking into CleanVar. CleanVar is a database that you have the SNP information associated with given diseases. And the y-axis here shows phyllopia score, which is a measure for the conservation. And then the benign variants shows more or less like a normal distribution. But if you look at pathogenic variants, we can see these are distributed in a way they have a shift or a skew distribution towards highly conserved regions. So it suggests that mutations in these regions may have more severe consequences. They can have a strong impact on the functionality of the genes. CAT is another score which mirrors the same picture. Again, it is a conservation. The higher value shows these regions are mostly conserved. So changes within this region can have a strong impact on the functionality of the genes. Multi-omics approach is also another solution that we can use in order to reduce the biases. Herring sex determination was one of the examples. One thing which I didn't tell you was we had the genomic data, we had RNA-seq data, and we also had proteomics data from sperm. So we could characterize the genes that we identify. And it helps us to have a better understanding, and also helps us to have a cross-validation across different layers. We have our finding at the genomic level. Let's see what's the picture in the transcriptome level, and also on proteome level, and so on. And it helps us to have a novel discovery by characterizing interaction between molecular components. Now I switch again and talk about something which is not scientific at all. Any question before we leave the science realm? Okay. Some of you might have experienced some of these, some may not. And if it happens to you, you are not alone. So it's part of the improvement as well, learning curve as well. We are all humans. We make mistakes, and in science it's also probably more likely to make mistakes. And we do science because we don't know the answer. So it's also possible to get non-significant results. But the community has formed in a way that we mostly hear or read successful stories and the publications. And the examples that I showed you was just to show you a bit behind the scene, what adjustment and correction we need to do in order to reduce the biases. Saying that, I give you a case study. This paper it was about speciation in rabbit and gene imprinting. I think it's the first time I'm presenting this. It was rejected 7 or 8 times. It was one of the papers which went into my thesis, and when I was doing that, there was a crash on the server, and I lost almost 1 year of my work, and I had to repeat everything in a very short period of time. So it may happen to you as well. Hopefully not, but if it happens, that's part of it. So to make the long story short, we have 2 subspecies of rabbits in Iberian Peninsula, Caniculus and Algirus. And they are distributed across this line that you see. These are the samples that we have collected. And the gray zone in between is a hybrid zone, where these 2 subspecies breed and mate. But the offsprings show low fertility. and we characterized it by finding sperm morph anomalies among the hybrids. So they had reduced fertility in males. We had genomic signals, very nice signals associated with abnormal sperm numbers and similar traits. So you would immediately say, maybe if anything is happening, it should be in testis, where spermatogenesis is happening. So we try to address this by looking into allele-specific expression. Do you know about this concept? If not, then I try to explain this through this picture. So some of the mutations happens in regulatory elements upstream of the gene. So in this case, we can see we have two mutations, two alleles, one C and one T, and these are linked or phased with another allele within gene body, which is here. So C comes with A and T comes with G. When we have C, it increases the transcription activity. So the transcription machinery can transcribe the gene. We see more activity and more expression of the gene. We can label it as active transcription. On the other hand, if we have the T mutations, it can limit the interaction between transcription factor, transcription machinery with the regulatory elements, and eventually the expression that we see in this gene. We can label it inactive transcription. So the idea of allele-specific expression is that to identify, based on the genetic profile, which genes show active transcription and which genes show inactive transcription. We had the genomic signal. We had a list of genes that we expected to see difference. But one of the key problems is that when you have working with wild rabbit, you cannot keep them in captivity. They do this suicide mission. They bang themselves into fences. So we cannot keep them as like a lab animals in the lab setup. So we had some samples, some individuals, and some parents as well. But we couldn't really say who is whose offspring is this individual or that individual. So it was very tricky. But it was not the only tricky part. We didn't find any striking results. In addition to what I just described you, so I was thinking what would be the explanation why we don't find any imprinting genes, or not as many as expected, especially in the regions that we had identified with genomic signal. I thought a bit more about the tissue that we are looking. So testis is where we have meiosis. We have diploid and haploid cells with the status of haploid and diploid states. And it's the place that epigenome is completely changes. We have chromatin remodeling, which can influence the expression of the genes as well. So it's like a soup of many possible scenarios. And also in human, the lowest number of imprinted genes has been reported in testis. So I went forth and back a lot of times, and then I reached my PIs, my supervisor, my team, and said, look, these are the biases that we have. And one of the reasons that we don't see the picture we expected is because of these biases. So with this, I would like to tell you that we are all human. PIs and supervisors they are also human, and we all base our reasoning based on the prior knowledge, but it may not be a complete picture. So if you end up in such situation, don't feel stressed. Try to collect your evidences, your reasoning, and then approach your team to convince, to motivate your decisions, and think about alternative solutions. Just to tell you one possible solution, which I never followed on that. I was thinking to write a grant application for that, but it never happened since I started working in Omics. But one approach is to try to do single cell sequencing. So instead of looking into like a soup of different cells, you can either do cell sorting. You say, okay, specifically I look into haploid cells or diploid cells, or do single cell sequencing. So you do not need to do any selection beforehand. You sequence everything, and then based on their expression profile, you can label different cells. Now, one thing which I would like to have your attention is when you are thinking about research conducted in different regions. So we are living in a world full of diversity. We have cultural differences, political differences, and it can influence the science as well, conducting the science and also accessibility. It was a recent publication in Science, an article showing that, countries in global north has a better access to science and the products of the science. COVID-19 is probably a good example. That countries in global south didn't have access as we had in the northern hemisphere. So we have these biases, which can potentially be influenced by political reasons as well. Another layer of the sort of complexity can be like the pressure that we have on publishing significant results, which may not give us the chance to read or hear about the failures or the adjustment we need to do for our research. And I gave you very few examples. There are more complex examples out there, and you may also experience it yourself. Speaking of that, you're in a level that you are learning new skills all the time. So it's not only you, we also who are like past, we are not sitting in those chairs anymore, but we are all the time, we are learning one way or the other, different skills. Has anyone read the book by James Clear, Atomic Habit? No, it's a really good book, and this has been inspired by that book. So this is the time on the x-axis, and this is the progress and results that you see. So the expectation is that as we spend more time, we expect to see better results, quicker results. But the true picture is that it's like exponential. So it takes a while to sort of see the expected results. And the gap between these two lines is called value of disappointment. So while you are going through this path, there is one element which was not in the book, I added myself, is that mistakes is everywhere and every time we do something. So like Adam Grant says, it's become comfortable with what you're uncomfortable. So mistakes is part of learning. So if it happens to you, do not give up, just move on, and that's part of learning. I wrote an article on my personal webpage as well, going through the value of disappointment and beyond. So if you're interested, you can read about this in more details. Shall I move on? So it's also important to keep balance between personal and professional growth. So it is what you see at that very moment, but if you zoom out, look at the bigger picture, you have gone a long way to reach here. So it's a fluctuative path, it's not a straight line. So it has happened to many of us and will happen again, but continuously adapt and learn. Sometimes we get this tunnel vision, we want to focus on the research and what we are interested to do, and sometimes we forget our human aspect of ourselves. So these are the books that help me to sort of keep this balance. Books by Adam Grant, Atomic Habits, the 7 Habits of Highly Effective People by Stephen Covey, and last but not least, Factfulness by Hans Rosling, which should probably be a known name for Swedish people. He was also living in Uppsala. He was a professor at KI. It's a really nice book. I highly recommend to read it. And one of the products of this book is Gapminder. There's a website which gives you data that help you to have a correct picture of what is happening in the world. So, take-home message. Think outside the box. The example that I showed you already explains that conventional approaches may not all the time give you what you expected. So think about like innovative approaches, alternative approaches all the time. Absence of evidence is not evidence of absence. And do not give up without a fight. It may be challenges, setbacks, just move on. And be kind to yourself before being kind to others. It helps you. And share the knowledge and positivity. It helps a lot. So with that, that was what I wanted to say. mix of both scientific lessons and also non-scientific lessons. I hope you find it useful, and I will be happy to take questions if you have any. Thank you.